go ahead and welcome everybody on behalf of GCC. We're really excited today to hear about a new core that was recently funded that will kind of build on some of the existing resources over at TMC Innovation Factory, specifically to support folks who are developing healthcare innovation within the device space. And so with that, I'm going to turn it over to Dr. Jason Sakamoto, who is the lead of this initiative. And turn it over to you, Jason, and you can tell us about this exciting new resource.
Well, thank you so much, Zan. And before we sort of get into the actual grant, we're gonna tag team this a bit with my colleagues here. So I'm going to turn it over to Devin Dunn, who runs our Accelerator program, and she's just gonna give an overview. And I believe then she's going to segue that to Jennifer, and she'll go over the ACT program to set up some context. So off to you, Devin.
Perfect. Good afternoon, everyone. Thank you for joining us. Really excited to share more about this new core facility, which has really been led by Jason. But just to give a little bit of context before we get started, I'm Devin Dunn. I'm our Director of Accelerator programs here at TMC Innovation. I'm going to go ahead and assume that everyone understands a little bit about TMC Innovation, and the role that we play in the ecosystem. But if we want to go to the next slide, we'll give a little high-level context. So TMC Innovation, the Innovation Factory, in our 10th year now, and really, as you may know, purpose-built to support entrepreneurs here in the TMC ecosystem. So whether that's a researcher in a lab with an idea, wanting to spin something out, whether that's someone who has caught the entrepreneurial bug, participated in a startup before, and now ready to found their own company, or you're a serial entrepreneur working on your next endeavor. We are that place and infrastructure to help support the entrepreneurial journey, kind of wherever you find yourself on that path. And if we go to the next slide. A little bit about how we do that. So broadly, when we think about entrepreneurial support, we think about that in a few ways. One is the physical infrastructure that companies might need, be that prototyping facilities, which we'll talk a little bit about, or lab space, which we have as well. There's the education and programming component. So that's where our accelerator programs come in. Some of you are likely familiar with our accelerator for cancer therapeutics, which Jennifer will refresh on a bit. We have our accelerator for health tech, our international programs, as well as our biodesign program. So structured programming to support entrepreneurs across medical device space, digital health, and therapeutics as well. The third piece of that is, of course, our venture funds. We think about the infrastructure, the programming, and the funding that founders may need, and work with our venture fund and our venture fund network to support founders in that way. So over the last 10 years, we've worked with over 300 startups who have both either been in residence with us or come through our programs, or both. We've founded 17 biodesign companies, also led by Jason, and very proud of the progress that all of those companies have made in going on to raise over $5 billion in total funds. So that's just a little bit of context. And when you think about the Innovation Factory and TMC Innovation of where we spend our time, which leads us into both kind of what we've done historically in terms of a core facility, which Jennifer will talk a little bit about, and how that's really led us to what is next with the Accelerator for Cancer Medical Devices. So Jennifer, take it away.
Thanks, Devin. So hi, everyone. My name is Jennifer Ruhi. I serve as the program manager for the Accelerator for Cancer Therapeutics. And so it's helpful for me to kind of give you a little overview of the program because as we stand up and build the Accelerator for Cancer Medical Devices, we will be using some of the structure and success that we've seen in the ACT program to kind of help build us out, a network and a curriculum that will be valuable for everyone. And so, again, program manager, definitely that person that's that bridger and connector for all of our participants. And so here on this screen, you're going to see just a quick about us. So program was launched in 2021 and it has run through until this year. This is our last year. We are currently CPRIT funded. So I'm not sure if all of you know, it's the Cancer Prevention and Research Institute of Texas, a $6 billion fund that has given us a grant to provide this amazing curriculum to all of our participants. We were also launched in collaboration with the GCC and the University of Texas Medical Branch. And so, again, our mission, I'm not going to read it to you all, but it's really to help build out the biotech ecosystem and to build, strong partnerships and relationships so that everyone that joins our programs here at TMC Innovation has access to the best talent, investors, and consultants that can really help push their programs to the next milestone. And so to the right, we've got some really great numbers that we're very proud to show as far as what we've achieved in the program. So we've had 103 cancer therapeutics projects that have been accepted from 19 participating academic institutions, as well as from all throughout the world and the United States. We've had 8 CPRIT product development awardees come through our program. 34 new companies have been created. 3 assets are in the clinic. 4 our leads have been generated with our computational core. And then very excited about the total dilutive and non-dilutive funding at just over $273 million. So next slide. So here you can kind of see Texas, huge state. These are all of the lovely institutions that we work with, and we have really great relationships with all of these technology transfer offices here in the state of Texas. And so, as you know, it's very important to have a great relationship with your TTO because they can kind of help guide you along as you're looking to commercialize. If you're coming out of academia, of course, but how to commercialize your asset there. And so, again, Houston, Third Coast, that's what we want to really push here. It's not just about the East and the West. There's a lot going on here in Texas. All right. And so here's, again, the snapshot kind of broken down for everyone. So $55 million has been secured in non-dilutive funding throughout the program. And then $218 million has been secured in dilutive funding. So very excited there. And ACT truly pushes those participants to reach those commercialization milestones. So again, very much about connections and providing you with the resources that you need to get the funding that you need to get into the clinic. And so now we'll kind of discuss some of the success stories. So I work with a team of entrepreneurs and residents, and so we really like to think of this as kind of our way of helping build the ecosystem. Pardon me, guys. It's cold season. So March Biosciences, they have been kind of in the news recently. We're very excited for them. They've raised over $50 million, just closed an oversubscribed Series A for $28.4 million last fall. So very happy for them. And so Sarah Hine, third picture in on your screen, was one of our first EIRs. And so she found really great synergy with Max Mamonkin with Baylor College of Medicine. And they decided to spin out March Biosciences. And so they're doing a lot. We're really excited for them. And then we've got Michael Torres, top picture on the left with Crossbridge Bio. And so he found some really great synergy with Shashan On, who is from UT Health, and they have currently raised 10 million in seed funding, and they're CPRIT awardees, and they're also going through CPRIT diligence. So very excited for them. And I'll hand it over to Jason.
All right. Welcome, everyone. My name is Jason Sakamoto. I serve as Associate Director for the Center for Device Innovation. I also head our TMCi Biozyme program, and I also serve at the pleasure of being the PI of this grant that's been a collaboration with TMCi Innovation as well as GCC. So working off of the great successes that Devin and Jennifer just mentioned on the ACT program, we wanted to see if we can do something similar, but in the medical device space. And this new proposal and this new opportunity will now be forward facing towards those developing devices, which is near and dear to my heart. Let's see. There we go. So here's a picture of our machine shop here within the CDI. As you can tell, we have a lot of fantastic CNC machines, just a state-of-the-art prototyping facility. We are going to be launching this accelerator in 2027. We needed a year to sort of set things up, get all the EIRs in place, and to make a capital purchase of a very specific 3D printer to help out with everything and create that core. So keep an eye out for probably applications opening in late 2026. Our mission, similar to ACT, is to do exactly what ACT did, but for medical devices. Let's create the next generation of cancer-facing med devices, and let's do that here. And so what this proposal, this grant opportunity offers is we have our state-of-the-art machine shop. We can offer advanced 3D printing capabilities. What I mean by that is going to be industry-grade 3D printing capabilities with micron-like type of resolution. So something that you don't find very often. We're also going to be offering computational simulation services. We all know in silico computational mathematics to be able to simulate through your prototyping process is a whole lot cheaper and more economical way to develop your next prototype. And then we're also offering to a select folks that are coming through our accelerator, we can support up to $7,500 with the prototyping support. So what does that mean? Access to our machine shop, our engineering machinists. We'll have a curriculum and mentorship program to sort of provide the entrepreneurship 101 to those going through the accelerator program. And in addition to that, we'll also probably provide some grant writing services, get that critical non-dilutive funding that really helps people bridge that gap. I sort of mentioned this already, but again, we're gonna offer to participants your startup 101. We're gonna have a whole network of service providers in the form of regulatory consultants or help with reimbursement understanding, all the important pieces and services to help you go to market. We're going to hopefully we are going to provide grant writing services to hopefully go after that non-dilutive funding that's out there. And again, hopefully very similar to what we do with our accelerator programs, we'll probably have access to investors and whatnot. So hopefully we'll have all this stuff ready to go in 2027. This is just going in depth and a little bit further of giving you some images of our 3D printing resources here on the right. Sorry for the collage. I just took those pictures with my phone yesterday. But on the right where it says 3D printing, on the left-hand side, that's our jewel of our J750 Stratus that allows you to do 6 different durometer materials at once. It's a fantastic piece of equipment. In addition to not only the fiber-based and the resin-based printing capabilities, we'll be adding another printer with higher resolution capabilities. And I did mention our machine shop already. In addition to more of the hard prototyping type of offerings, we have the in silico offering as well. We're planning on providing computational simulations core built around the COMSOL multi-physics platform. It allows you to do anything from fluid flow, heat transfer, and structural mechanics. The process. It's kind of corny, so bear with me. But basically, how we envision this is you come in with an idea. You'll be hopefully you'll have your IP all locked up at your respective tech transfer offices. You'll be coming in. You'll apply to the program. You advance through the selection process, and once you get admitted, you'll receive that expert mentorship from our subject matter experts and our entrepreneurs in residence. That's what ERR stands for, to help guide you through that commercialization process. Being an academic myself, I understand that sometimes we don't have the full picture as an academic. And so going out and having a place that will consolidate this type of knowledge will truly be meaningful. We will have commercialization workshops, something we do very well with our experience with running accelerators over the years. We'll provide funding for prototyping so that you can work inside our machine shop while using our machinists and engineers. And you'll also have access to that computational core that I mentioned before. So here's the team. It's pretty much many of those that are on this call, and then our fearless leader, Tom Luby, on the top left. That kind of consists of the team. Here are our external advisory committee, and as you may recognize these faces anywhere from Billy Cohn to Dana Deerdorf and Justin Bird. A lot of clinical as well as engineering expertise on this board. So I'm very happy to have them in our corner. So here's my contact information. But before we go, what we'd really like to do is introduce Tushar Sharma. He's the CEO of Vivify. And we really wanted to demonstrate and give you an example of a company that was born in the CDI, leveraged the CDI resources, both from a machinery standpoint and an engineering side, and so I've asked Tushar, who's a good friend of mine, to kind of go over and give his journey, his experience, and what he's been able to do and benefit from the resources in the CDI. He may or may, I think he's cancer curious with his technologies, but it may not be a perfect example of a cancer type technology. But I did want to bring someone in that has used our facilities for many years now and has taken his device to just extremely insane levels. So with that, Tushar, I have your slides. You just kind of cue me in and I'll start them up. I'll go to your first one now. Are you ready to go, Tushar?
Yes. Thank you so
Fantastic.
much, Jason. It's a pleasure to be here, and I've been at CDI as a facility since 2019, and it's been a tremendous opportunity for us to leverage all of the amazing infrastructure that CDI has to progress Vivify through various phases. So what we are doing is we are developing a completely novel approach to treat prostatic conditions, both benign and cancerous. We are starting off with the benign tumors, which is benign prostate hyperplasia, and then prostate cancer is our subsequent indication for use for our device. So let's jump into just real quick on the next slide. We have what benign prostate hyperplasia is. We will play the video. So BPH, or benign prostate hyperplasia, is a very ubiquitous problem. More than half of all men above the age of 50 are affected by it. It's the condition where benign tumors start to grow in the prostate, and as a result, it starts to constrict the urethra, and therefore men cannot empty their bladder in a single go. And this forces them to wake up multiple times in the night to force empty their bladder. Now we have known for over a century that testosterone is what causes growth of prostatic tissue. But looking at the human physiology, testosterone is produced by testicles in the human body and drained through the venous side. It's dumped on the venous side as a byproduct. So these veins can carry up to 150 times higher testosterone in concentration compared to serum levels. And one of these vessels the differential vessel takes this testosterone-rich blood straight to the prostate from the prostate, which leads both benign and cancerous conditions to manifest over time. So if we go to the next slide. So when we really started working on this problem, we wanted to do a vascular replumbing. If you click one more time, Jason, that should activate the animation. What we intended on doing was replumbing the pelvic floor vasculature to make sure that none of the testosterone-rich blood ever makes it to the prostate. And it involved two key critical steps. One is connecting the spermatic vein, which is the primary drainage route for testicular venous blood, to an adjacent blood vessel so that it doesn't have to go through all the level to the level of kidneys before it goes into the systemic circulation. And then the other step was to ligate or close off that shared blood vessel between the testicle and the prostate. So the genesis of this particular concept and this particular procedure was in 2019 when we actually started working on contemplating of doing this kind of a procedure. And so in the next slide, we started with working on the potential device that could do the hardest step, which is connecting the spermatic vein to an adjacent blood vessel. And since urologists are the ones who owns the BPH patients, we said, okay, what kind of modality can urologists do? And laparoscopic and robotic is what jumped to the top. So we said, great, can we come up with a device that can connect two blood vessels through a laparoscopic approach or through a robotic approach? And these blood vessels are tiny vessels. We're talking about 2 millimeter to 3 millimeter veins, which are super flimsy and really hard to manipulate. So over the years, we created and started working on the device. And on the next slide is the latest version of the laparoscopic device that we ended up creating. And all of these components and the end effectors and the more impressive mechanisms and components that go into the handle part of this device, which you won't be able to see in this particular set of images or the video on the next slide. All of them were prototyped at CDI. All the components were prototyped and machined at CDI and assembled at CDI as well. The way it works is we have this tool called a vessel diverter, which goes through the first stapling ring and attaches to the end of the first blood vessel, and then pulls it through the stapling ring, and then flares it out and transfers it to the stapling ring. Then we pivot this entire diamond shaping cage to repeat the same procedure for the second blood vessel that we want to connect to the first blood vessel that's already loaded on the device. So once we have the second blood vessel loaded on the device, we can then essentially close the entire cage or the diamond-shaped structure, and those needle pins in those stapling rings press fit with corresponding mating holes in the other stapling ring and permanently and irreversibly connect the two blood vessels together. This was the first time anyone has been able to do this on such a small scale from a laparoscopic approach. And all of the components that you see over here are prototyped at CDI in the machine shop at CDI. So that was the first part that we were working on. And then in the next slide, there's a video of a evolved prototype of the same thing in an actual acute animal lab. So similar concept where we have the everter that goes through a coupling ring and transfers the vessels onto the stapling ring and eventually closes the two stapling rings and connects them together. So this whole thing was done laparoscopically, and you can see the two blood vessels connected with each other in a live animal. And then other step in the procedure was to close off that shaped blood vessel between the testicle and the prostate, right, which is the differential vessels. And so the way we envisioned that was having a laparoscopic device that can just clamp around the vas, which carries the sperm in males all the way from the testicle to the prostate, and the tissue around the vas is what carries that testosterone-rich blood over to the prostate. So if we can grab hold of the vas and the surrounding tissue and have a conforming device that can do a circumferential ablation around it, we can prevent testosterone-rich blood going to the prostate and therefore reverse both of these benign and cancerous conditions. So this was one of the prototypes that we developed in the CDI facility again. Going into the next slide, there is an actual image of the prototype on the right, and then going into the next slide, there's a image of the device being used in a cadaver lab. And so we successfully tested this out, and I think it took us less than 2 weeks from the concept to actually prototyping it in the lab to getting into a cadaver lab and testing it out. And that's the beauty that CDI offers, because pretty much anything that you can conceive of, you have the resources and infrastructure to build it out over there. And so the next thing that we had to do was to show whether this procedure works in humans or not. So on the next slide, we were the first ones to propose this vascular mechanism and the new procedure to reverse all of these conditions. And so we had our surgeons from UCSF and other places fly down to Panama and do this procedure open surgically, where they anastomosed or connected the spermatic vessel to an inferior epigastric vessel, and then ligate of the differential vessels that go along the vas. And the outcomes from these procedures were actually phenomenal. At 3 month follow-up of these procedures, we were seeing symptom improvements and prostate volume decrease that was much more significantly better compared to what medications would show at 6 months follow-up point. So twice as effective in half the time frame. But there was a thing that we realized was in some of the cases, we could not complete the anastomosis procedure, and they still showed improvement. And so that led us down the pathway of, hey, could we actually do a simpler procedure? And this pivot or the simplification of our procedure and our technology, therefore, is a very recent one. So we're talking in the last 2.5 months. We decided that, hey, can we actually do a simpler procedure? And yes, if we can do a simpler procedure, can we make it less invasive compared to a laparoscopic procedure? Can we just do it non-surgically, for example? How less invasive can we get? And so that led to the next prototype on the next slide, which is our latest concept and device that we're working on. It's a non-surgical device that accomplishes ligation of the vasculature around the vas without any incisions. It uses a needle array to deliver bipolar energy, and only a few needle punctures remain at the end of the procedure. And these needles are really, really tiny. We're talking about 3 times the thickness of a human hair. So really tiny, as close to in minimally invasive procedure or non-invasive procedure as one could ever ask for. And all of these prototypes were tested at CDI, developed at CDI, assembled at CDI, and tested in 10+ cadaver labs in less than 3 months time span. And that is where what we are working on right now. So in the next slide, it shows a video of how the device works. I should warn everybody that there's explicit anatomy over here. Now we are going through a scroll approach. Just like a vasectomy, we grab the vasal packet, which holds the vas along with the differential tissue around it manually, and then use our device to clamp around it. And then we have all of these 3D printed cartridges which have needles in them that can be advanced. So we insert the cartridges into the device, advance the needles, and those needles essentially surround the vase in a concentric manner. And then we connect all of these needles to bipolar generators and just ablate it. And as you can see from this, when we're ablating them, the tissue dehydrates, it collapses the vessel and squeezes all the fluid to the top. And in less than 5 seconds to 10 seconds, we can essentially close off the entire tissue. So going from a really highly complicated technology, which is laparoscopic technology, with with more than 1,000 parts that went into assembling the device, and then switching gears to going to a non-surgical device, and then taking it from concept through rapid iterative process to an actual device, which is ready for acute animal testing in less than 3 months. All of that is only possible because we have the resources and the capabilities that we have at CDI, and not to mention amazing mentors such as Jason and Billy Cohn and all of the folks that help us guide through the process and make sure that we avoid some of the pitfalls based on their experiences and based on what they have witnessed. That's all I have, Jason.
Fantastic. I guess we could, thank you so much for sharing that. Let me stop sharing. Let's see, share, stop sharing. Thank you so much, Tushar. That was an amazing story, especially, having such complex devices. The first one and the second, they're extremely complex, not easy to build, and it's just amazing you were able to do that. Your engineering team is just second to none. So kudos for that. Whoop, I think let me turn off the video. But anyways, so Suzanne, do we now go into the Q&A portion of the day here?
Yes. So we are in the Q&A, although I have noticed that there have been questions being entered into the Q&A throughout the whole thing, and Jennifer's done a great job of responding to all of those. We did this as a webinar this time. I don't know if you remember, I don't think you were on the call, but last year it was our only one that's been bombed by somebody that really messed with us having this as a meeting. So we don't have the, the option this time for people to unmute their mics and ask questions, but we had some sent in advance. And so you can address some of those, and we'll leave the Q&A and chat open if anybody wants to enter questions there.
Suzanne, I don't see anything in the webinar chat. Am I missing something?
Well, if you look at the Q&A, that's where it looks like there's one that's open. It's just a comment to Jennifer, but under answered, you'll see all the ones that Jennifer has responded to several. And then there were those that were, so if for those that are on the call, please feel free to continue to enter any questions in the Q&A, and we can address those live. And in the meantime, Jason, if you'd like, you can look at some of the ones that were submitted in advance. I think that Dawn sent you a few of those.
Sure, so I think the first question that I saw was, what areas are priority? And so I would have to say we're pretty agnostic, other than it has to be in the cancer space. When we were writing the proposal, we looked at all the NCI grants awarded within Texas, and anything that could be that could utilize or did utilize medical devices, I put that in a bin and we did an analysis. And what I've seen was that there was a lot of requests or technologies that dealt with microfluidics. There were a lot for cell sorting, organ on a chip type of plays. And so that is why we felt that it was beneficial to the community if we're able to get a high resolution 3D printer that can do all the above there. But that doesn't mean that it has to be fit into that box. We're open to anything as long as it's a medical device and it's facing cancer. Jennifer, would you like to add on anything on that?
Nope, you covered it.
All right.
Yep.
We do have a question that just came into the Q&A that said, what is the transition for existing J&J participants with this new core program. So presumably this is talking about anybody in the device space, any opportunities there?
Let's see, so are we, I didn't quite understand the question. I did hear J&J for those that are transitioning from J&J.
That's what it says. If whoever entered that question, if you would like to clarify it, that'd be great. But what it says is, what is the transition for existing J&J participants with this new core program? I mean, they're completely separate, but I could imagine that there might be some opportunities. Anything you'd like to weigh in on that? I mean, they definitely are two totally different programs, but
Well, maybe it's from J Labs, and because that's a lot of therapeutic focus over there, and
right.
some of them are cancer. If there is a utility for a med device, it doesn't have to be your final product, but if you need some sort of a device to help finalize your product, maybe if you need optimization of ligand binding analysis or something, and you need some device to help you with that, although your final product is not a medical device, that still counts because you still need some sort of assistance in prototyping to help you get there. I hope that was a good enough response for you. If any of my colleagues would take a different interpretation of that, I'm happy to open the floor to them.
Yeah, I think you covered it, Jason. Maybe the one thing I'd add, you could be referring to the news that came out a few weeks ago that TMC will be more involved in J Labs going forward. So there is a transition there. I think one of the great parts about that transition is we'll be able to facilitate more synergies between what J Labs is and what CDI is. So that overlap between particularly kind of the medical devices within J Labs who might benefit from access to CDI facilities will only become easier, which will be great.
Thanks, Devin.
Yeah.
There's another question here about do overarching agreements already exist between the new core and the Houston Methodist Research Institute organization? No, not yet. There's no formal agreement between the RI and this new proposal. But having lived at and worked at the RI for 5 years, we have really good connections there. So if something needs to happen, I'm sure we can work things out. But there is no currently any formal relationships or agreements in place between ACMD and Houston Methodist Research Institute. All right, what are the ideal stages of development for startups to participate in this accelerator? That's a really good question. And I think similar to how ACT handles things, where we welcome all comers. As long as you're in Texas and you're investigating or you're in the cancer space and specifically leveraging medical device technology, I think we're open. I think we can meet you to where you're at. Many folks may have a very limited understanding and how to translate things, and that's what we're here for. But also, if you're there and you have you're a little further along, we can meet you there as well. So I think we're pretty agnostic around the product development pathway. Jennifer, any other tidbits on that you'd like to add?
No, you're doing great.
Oh, who would have guessed that? The final question I have is any available job opportunities in the medical device space? Man, it's tough. It's a difficult time right now. We do have 17 companies that are leasing space within the Center for Device Innovation. I would keep an eye out on when those companies are raising, and typically when they do raise a round, they're always trying to expand their team. But right now, I don't take an ongoing tally of who's hiring at the moment, but we do have a folder of resumes for those looking for different opportunities that our CDI companies have access to. So when they are looking for new team expansion members, they have a place to go to.
I'll also plug our Innovation Factory newsletter. You can go ahead on our website and sign up for that if you don't receive it already. We do post job postings in that newsletter for the companies that we work with. So good place to check out.
Jason, I'll go ahead and ask a question because we've had a number of non-cancer device projects come through Reach. Can you just talk a little bit separate from ACMD, how folks can engage with you and your team and others in that space as kind of a follow-on?
Sure, I'd be happy to. So there are a couple of ways that you can leverage the CDI and its resources. The easiest would be if you lease space here. Those companies that do lease space, they get 4 free hours of machine shop time a month that can be used for actual machine shop time or engineering machinist time. And then we only charge $50 an hour after that, which is well below market. Another way to gain access to that is through, we have a CDMO that works out of the CDI. It's called Merge Medical Studio. And so we love that relationship because those from different institutions can quickly create a fee for service type agreement there. All the IP stays with the inventor, and they have access to our machine shop engineers and so forth. And they're very reasonable rates. They're probably at market or below. So those are the easiest ways to gain access to the resources at CDI at the moment.
All right. Thank you, Jason. I don't see anything else. Any final thoughts?
Well, from my perspective, I'm super excited to get this going and working with you, Suzanne, and your team. I think we're especially excited knowing the success that we're building off of from the ACT program. So yeah, I was always jealous that the med device folks didn't really have a thing, and now we do. So let's go and let's go do it.
All right. All right. Well, thank you. Thank you to Shar and Jennifer and Devin and Eliana. I didn't hear too much for you, but good to see you as well. Yes, we'll try to get some resources for the website. And, if anyone has any additional follow-up questions, feel free to reach out directly to Jason and his team.